therapeutically useful small molecules. Dr. Mo, welcome. The screen is yours. There he is. All right. Dr. Mo, you are unmuted.
Excellent. Thank you.
You are welcome.
All right. Can everybody see the presentation? Great. Okay, well, thank you very much for the very kind, very generous introduction, Pete. I want to start by, again, thanking Pete and Michael Gilmore for inviting me to speak today. It's really nice to be part of the MSI Annual Symposium this year and reach such a large audience, actually. I want to thank everyone that zoomed in on YouTube, and a special thanks to my MSR and Harvard Medical School mentors, Roberta Coulter and John Clardy, and the entire Clardy lab, who I know is following this live as well. Okay, so my lab is in general interested, as Pete mentioned, in the discovery and characterization of secondary metabolites or natural products from microbial sources. These are small organic molecules synthesized and secreted primarily by plants and microbes into the environment, where they then carry out a myriad of functions. And we are especially interested in the roles of these small molecules in mediating microbial symbiotic interactions. These molecules represent a kind of a chemical language with which microbes communicate and compete with one another. There's a great effort both in my lab and elsewhere to decode that chemical language. We're also interested in the therapeutic uses of secondary metabolites. Penicillin listed here is a well-known secondary metabolite. We heard a lot of other antibiotics in Elizabeth's talk. Those are also all of almost all of them of microbial origin. And so with every discovery of a new secondary metabolite comes at least the potential for therapeutic utility, which we try to explore as well. And lastly, we're interested in how these molecules are synthesized. This leads to new microbial chemistry, new biosynthetic pathways, enzymatic reaction mechanisms, and that's an area that we're engaged in as well. Now, secondary metabolites are genetically encoded. These arrows that you'll see throughout my talk represent genes, sets of contiguous genes that we call biosynthetic gene clusters. And the enzymes that they encode then assemble complex molecules like penicillin in a stepwise fashion from relatively simple building blocks. Okay, so when we talk about metabolites or metabolism, what comes to mind is probably something like this image. Primary metabolism, so all the molecules that are inside of the cell and allow bacteria, microbes to grow and divide. And work over the last century really has given us a really detailed, quantitative, holistic picture of primary metabolism. This kind of understanding is not yet available for secondary metabolism, so all the molecules that are primarily or for the most part outside of the cell. But with recent advances, we've started to ask, can we achieve a similar understanding? And what are the challenges involved in doing so? And I think a comparison to primary metabolism is helpful here. So primary metabolites, as I briefly mentioned, are essential for growth and proliferation. As a result, the pathways are always on, TCA cycle, glycolysis, for example. And many of the enzymes are robust and present at near millimolar concentrations inside the cell. That is not the case for secondary metabolism. These are not required for growth. And most pathways are, in fact, not on, referred to as silent or cryptic. And to achieve a similar understanding, at least we need to be able to turn on these silent or cryptic biosynthetic gene clusters. A second key differentiator is primary metabolism consists of a plethora of interrelated intermediates, as you saw on the last slide, and this helps immensely with isotope tracer studies or finding structures of new intermediates. This, again, is not the case for secondary metabolism. Many pathways don't even have diffusible intermediates. And for the most part, one pathway is not at all related to the next one within a cell. And so the picture that this leads to is one where primary metabolism is specific. It converges to a few well-known end products. Secondary metabolism, on the other hand, is highly divergent. because of the different pathways, distinct pathways, and it leads to many unknown end products. And so in my mind, at least, the two main challenges in achieving a similar understanding of secondary metabolism include turning on the silent or cryptic biosynthetic gene clusters and figuring out ways to structurally and functionally characterize all these divergent secondary metabolites. Okay, let me elaborate a little more on this aspect here, the silent nature of most biosynthetic pathways. I think one of the examples that demonstrated it to the scientific community was that of erythromycin. Here's the structure of erythromycin. It's a natural product or secondary metabolite that's been used as an antibiotic since the 1950s. We have second and third generation erythromycins like azithromycin or Z-Pak. Even today, this natural product is not synthesized, but isolated from giant fermenters growing this bacterium, Cyclospora erythrae. We know a lot about this actinomycete bacterium, a lot about erythromycin, and ultimately researchers in the field identified the biosynthetic genes that are responsible for its synthesis. But the real surprise came when the genome sequence of Cyclospora erythrae was determined. And what it showed was not just the erythromycin gene cluster, but another 20 or so biosynthetic gene clusters that had no products associated with them. So here we were as a community growing this strain on massive scales, really all over the globe for several decades. And we had not anticipated that it had the potential to make another 20 or so secondary metabolites. These are what we now call silent or cryptic biosynthetic gene clusters. These are gene clusters that you can clearly identify in the chromosome of a bacterium, but that for largely unknown reasons are not actively expressed under standard lab growth conditions. And so as additional sequences accumulated, it turned out that these silent clusters are abundant, that they actually outnumber those that are constitutively expressed. And this is a small table to make that point. It could be a lot longer. I won't go over the individual strains, but basically what it shows is that many of these prolific producers have more than 20, 30, 40, 50, sometimes 60 biosynthetic gene clusters and a corresponding capacity to make that many secondary metabolites, but only a small fraction of these are actually leading to the synthesis of detectable levels of secondary metabolites. And so this is one of the key challenges that we decided to pursue when I started my independent position at Princeton, to try to understand how these gene clusters are regulated and how they might be activated to find new secondary metabolites. And we've done this with several approaches. I'll focus on one of these today, high-throughput elicitor screening, or HITES. Okay, so what are the challenges in activating a given silent gene cluster? The way we thought about this problem was that there are sort of three unknowns in this equation. You start with a completely uncharacterized biosynthetic gene cluster. You can observe it in silico, but really there's no experimental assessment of it. Presumably there's this unknown signal that can activate it. And if and when it's activated, there is a new natural product that needs to be characterized. And so how do we deal with these three different aspects? Bioinformatics has come a really long way in the last 10, 15 years, and really no one's scared of uncharacterized gene clusters anymore. In fact, people seek them out. So this is not a major concern. We've had for decades now old and new methods as well to identify and characterize natural products, and I think this is not a major concern either. The real unknown in this question is the signal. I don't think it's possible to look at a gene cluster and predict what signal might activate it. And so we wanted to develop a method that would essentially address this process, that would identify a small molecule activating signal or elicitor for any given silent biosynthetic gene cluster. And the method we utilized was a simple one. We would incorporate a reporter gene inside of the gene cluster that we were interested in. This would now give us a rapid readout for its expression. And then we could screen hundreds of conditions to see just what it took to activate these silent gene clusters. And so to test that idea, we chose a model organism and a gene cluster. We chose Burkholderia thailandensis, which is a non-pathogenic model for these bioterrorism pathogenic agents. It also has many silent gene clusters and is genetically tractable. Within B. thailandensis, we chose the Malleilactone or Malleilactone gene cluster. This is a cryptic virulence factor It's required for virulence and only activated upon infection. And because of its role, it had been heavily studied by the Brady and Hertwig groups, and the product had been characterized. And so it provided a nice proof of concept case to us. And so we either generated or obtained the proper genetic construct and then carried out high-throughput sequencing to see what it would take to activate this gene cluster in the lab, not in an infection model. And so here's the first set of results that we obtained. I'll quickly walk you through it. On this axis, you have the level of expression of this silent malleilactone gene cluster. Each of these bars represents the level of expression in the presence of a molecule from the small molecule library. These are shown in groups of 16. We have a positive control, we have a negative control. Now, without sophisticated analysis, I think you can see that some of these conditions activated this silent gene cluster, the two best shown in red, the next best two in orange, and then the next best 5 in blue. Now, what was surprising was the identity of the elicitors. The best ones turned out to be piperacillin and trimethoprim, both of these clinically used antibiotics. The next best 2 were members of the cephalosporin group of antibiotics, ceftazidime and cefotaxime, both also clinically used. And then the next best 5 were all members of the fluoroquinolone group of, again, clinical antibiotics. All of these antibiotics kill Burkholderia thaiendensis at high concentrations, but at sub-inhibitory levels, at sub-lethal doses that we use in the screen, they had sort of an opposite effect. They stimulated metabolism rather than inhibit growth. And so from a relatively small molecule library, we identified a number of elicitors, all of these clinical antibiotics that are toxic to Burkholderia thaiendensis. Now we want to verify that they were actually activating this silent gene cluster, and we did so using qPCR and metabolic profiling. Using qPCR, we could see a 5 to 10-fold activation of this gene cluster of the genes shown here in blue. With piperacillin, we could see an 8 to 45-fold induction with trimethoprim, very much consistent with the screening results. More importantly, and this is where having an identified product came in handy, we could directly monitor production of the secondary metabolite. This starred peak corresponds to malleilactone, and we could see that the control produces none, whereas piperacillin and trimethoprim now lead to induction of the secondary metabolite, as read out by HPLC-MS. And we estimate at least 15-fold overproduction with piperacillin and at least 150-fold overproduction with trimethoprim. So the method, at least in this proof of concept case, worked well, and we've been active in developing it further and applying it to cases to previously uncharacterized silent gene clusters. And one of those is shown on this slide. This is a large uncharacterized gene cluster at the time, Streptomyces albus. It encodes 4 large non-ribosomal peptide synthetases, all of these driven by this silent promoter, which we label p-SIR, and we call this the SIR gene cluster. This is silent under normal growth conditions in Streptomyces albus. And in this case, we showed that a triple EGFP reporter construct would work well in this organism. And I won't go over all the genetics, but we generated 2 constructs, reporter constructs, one with a triple EGFP reporter cassette driven by a p-SIR, the silent p-SIR promoter was inserted into a neutral locus, and another where we generated a site-specific insertion of this reporter cassette directly replacing the SIR-E gene. And so we then applied again high-throughput elicitor screening. In this case, we identified etoposide and ivermectin as the best elicitors when comparing the 2 different reporter constructs. Etoposide is a well-known anti-cancer agent. It also inhibits bacterial growth. Ivermectin is a very well-known anti-parasitic agent. So another example of low-dose toxins eliciting secondary metabolism, in this case in Streptomyces albus. And then using these elicitors, we identified the products of this gene cluster. We ended up characterizing 50 new cryptic metabolites. It ended up having a fairly divergent output. One of the major products were these surrogamides. These are octopeptides, cyclic octopeptides with a number of D and L amino acids. We identified 4 new analogs in this family. We identified the acyl surrogamides. These also have the same octopeptide backbone, acylated on the lysine side chain with a butyryl group. We identified the albucyclones, again, the same octopeptide fragment, now containing an unusual isoquinoline quinone acyl moiety. And lastly, we identified these linear decameric peptides, again, with a number of D and L amino acids and an unusual beta-alanine disposition. Interestingly, we found that SIR-A and SIR-D were synthesizing these octapeptides, whereas SIR-B and C were synthesizing this linear decapeptide in an independent fashion. An unusual example of one cluster generating two sort of different products. Okay, so we've applied it to a number of other cases as well, but really what was slowing us down was the genetic procedures that we had to use, especially in the actinomycetes, to get going. And so in the two examples I showed you so far, the workflow we used was the following. We would start with a wild type strain, carry out genetics to insert reporters or generate deletion mutants, and then use the high throughput elicitor screening with the genetic reporter readout. And so this is really the slow step. And we started thinking, how can we eliminate this slow genetic step? And with advances in mass spec, we started thinking, can we replace genetics with mass spectrometry as a readout? And in this hypothetical workflow, we could start with a wild type microorganism, no genetics, and then directly read out these induced metabolomes in each of these wells using some kind of a mass spec approach. And so if we use HPLC-MS with 500 or 1,000 conditions, this would probably take around 4, 6, maybe 8 weeks, and it would kind of be as slow as just doing genetics. On the other hand, if we could image these wells somehow rapidly, that would expedite our workflow. And so we became intrigued by this idea, and I, at the time, sort of bit the bullet and bought an imaging mass spectrometer for the lab. We became really good at using it to characterize these metabolomes, and now we can read out 500 or 1,000 of these induced metabolomes in one afternoon. That has really enhanced our platform. And so once we convince ourselves that we can use imaging mass spectrometry and this approach properly, we applied it to a first real test case. And this real test case was a rare actinomycete strain, an Amycolatopsis strain. We chose it for several reasons. One is a rare actinomycete. It is, at least for us, genetically intractable, so genetics was not even an option. Two, PCR screening approaches had shown that this organism encodes a silent vancomycin-like gene cluster. And vancomycin is a glycopeptide antibiotic, and so we thought perhaps we can activate its production using our approach. And so we subjected this microorganism to elicitor screening, and then read out all these induced metabolomes in each well using an imaging mass spectrometer. And I'll briefly show you sort of our direct readout. For example, with Elicitor 1, we might see this kind of a mass spectrum. And then with Elicitor 2, a little different. And then 3, 4, 5, so on and so forth, all the way up to 500. And then we amalgamate all of these individual mass spectra to generate a 3D plot that relates the M over Z, so the mass of the compound that we detect, as a function of its intensity and the elicitor library. So it basically provides a readout of the metabolome under 500 different conditions in this case. Now, a couple of things about this plot. We're now activating silent gene clusters in a high throughput fashion, whereas the genetic readout allows us to monitor the expression of one gene cluster at a time. This method allows us to see all metabolites that we can detect with this mass spec method. And as such, this is sort of an image of the global secondary metabolome of this microorganism, again, as read out or mapped by imaging mass spec. And as I mentioned in the outset, this is one of the major challenges in natural product, in the natural product field, figuring out the global secondary metabolome of a microorganism. And this might be a method that could help us get there. Okay, so is the elusive vancomycin-like compound encoded in this sort of mess of metabolites? We screened these for the chloride mass ion signal in the major compound groups that we detected. And of course, we did not see it. So either the compound was not being activated or we were in the wrong mass range. And naturally, we thought it was the latter. And so we screened in a higher mass range between 1,600 to 2,000 Daltons, roughly. And what we observed is one signal at 1811 that seemed to have this chloride substituent, which is very often a part of the glycopeptide antibiotics like vancomycin. If I take a 2D slice out of this 3D plot, you would see that for the M/Z1811 signal, the two best elicitors are dihydroxychristine and vincamine. These are both plant-derived alkaloids, both toxic to this bacterium. But at low doses, they seem to be inducing the production of this compound. We could show that in flask cultures, our DMSO control produces none of this compound with vincamine and DHE. Again, we get significant production of this metabolite. We then generated production cultures, isolated the compound, solved its structure, and it is shown here in its full glory. This turned out to be a new glycopeptide antibiotic. We call it Keratinimicin A. Just to briefly explain the structure, it has a 7-mer peptide backbone, just like vancomycin. It also has these unusual aromatic crosslinks, much like vancomycin. And it has some unusual sugar substituents like this actinosamide sugar that's limited to the Amycolatopsis. The stereochemistries of the sugars was derived from NMR. Those of the alpha carbon backbone are bioinformatic predictions after sequencing the biosynthetic gene cluster for this compound. We could also identify additional analogs containing additional unusual modifications. For example, this acosamide sugar in variant B, or this acosamide sugar and this glyoxylyl group in variant C. So we successfully found this elusive vancomycin-like compound, but one thing we were not happy about was the somewhat tentative assignment of these absolute configurations in the peptide backbone. And so we decided to take a X-ray crystallography approach, managed to really by luck crystallize this compound and solve a crystal structure, and the structure is shown here. And really it showed us two things. All the absolute configurations we predicted on the last slide turned out to be correct based on the biosynthetic gene cluster. Moreover, for the connoisseurs in the audience, this formate group here identifies the exact same position as the carboxyl group of the D-ala dipeptide terminus in the corresponding vancomycin structure, suggesting a similar mode of action, a similar target as in vancomycin. Okay, so we weren't quite done here yet. When we screened this sort of extended mass range metabolome further, we found two other much less abundant metabolites that also appear to contain chloride. In fact, if you maybe squint at this, you might even be able to see the chloride mass ion signature. We again generated production cultures of these, also solved their structures by NMR. And these turned out to be a new chemotype within the vancomycin class or glycopeptide class of compounds. They now contain a 6-mer backbone and have this unusual oxazolidinone cap at the end terminus shown here in blue. We call this compound Keratinicyclin A. And again, here we identified two additional variants. Okay, so what about the bioactivities of these secondary metabolites? Given the structural similarity to vancomycin, we thought these could be strong antibiotics. And that, in fact, turned out to be true. Keratinimicin A turned out to be fairly potent against a number of gram-positive bacterial pathogens that we investigated, with MICs in the 0.1 to 4 micromolar range. Notably, it had a 5-fold better MIC than metronidazole, which is currently used to treat C. diff infections. It was not active against vancomycin-resistant Enterobacter, again pointing to a similar mode of action as vancomycin. Keratinicyclin B, this unusual compound that we also found, unfortunately had no antibacterial activity. But we screened it against a number of viruses, and we saw both potent and specific antiviral activity against the human pathogenic virus RSV. This is especially relevant in newborns. And it turned out to be 20-fold better than ribavirin at killing the RSV virus. Ribavirin is currently used to treat RSV infections. And at 10-fold the MIC, we detected no toxicity against human cells, suggesting that this might be an attractive drug lead for future studies. Okay, so the examples I showed you have sort of one aspect in common, and but there are also a lot of differences. The organism differs in each case. The elicitor is different. The secondary metabolite that is produced in response to the low-dose antibiotics is different. We don't know the ecological rules of these compounds yet. We have some idea about their bioactivity in the lab. What they do have in common, though, is that low-dose antibiotics are eliciting the production of these compounds in each case. And in each case, these compounds, the elicitors, at high doses are toxic to the producing bacterium. So trimethoprim kills Burkholderia at high concentrations. At low doses, it elicits this secondary metabolite. That the effects of antibiotics are highly concentration dependent has been known for some time, and that they have effects that go beyond growth inhibition has also been known for a long time. But that they activate silent gene clusters, that is a new dimension of the idea that antibiotics can have effects other than growth inhibition. And so in the last few slides, I want to sort of touch on the mechanism of how low-dose antibiotics might elicit secondary metabolism in bacteria. I can tell you off the bat that this is a case-by-case mechanism. We have not yet found or examined enough of these to come up with sort of broad themes or general themes that apply. Okay, and this case that we have characterized in some detail is trimethoprim and Burkholderia thailandensis. It is well known that trimethoprim inhibits dihydrofolate reductase, a key enzyme in a one-carbon metabolic pathway in bacteria. Inhibition of DHFR leads to amino acid starvation, and this triggers then the stringent stress response via RelA and SPO-T and the secondary messenger PPGPP, which binds to RNA polymerase and alters its affinity for certain promoters, upregulating, for example, amino acid biosynthesis, downregulating other processes like replication. So the first question we asked is, does trimethoprim in Burkholderia thailandensis also act through dihydrofolate reductase, and is the stringent stress response involved? And the answer is yes and no. It does act through dihydrofolate reductase. When we treat Burkholderia containing a trimethoprim-resistant copy of DHFR, we find that in this case, trimethoprim does not elicit the malleilactone gene cluster as seen in a malleilactone LAC-Z reporter construct. The construct in the absence of this drug-resistant allele does induce the malleolactone gene cluster, as I showed you earlier. So even low-dose trimethoprim has to partially inhibit DHFR to exert its effect. But in this case, the stringent stress response is not involved. When we generate RelA and RelA SPO-T mutants, both of these can trigger malleolactone production as shown in this red peak. So what then mediates the response of trimethoprim? Because of the effect on this one carbon pathway, we focused or zeroed in on it. And what we did was measure by transcriptomics, proteomics, and metabolomics the levels of all the mRNAs, enzymes, and metabolites shown in this pathway. And what we found was the following. The way Burkholderia thailandensis responds to low-dose trimethoprim is by inducing the very first gene and the very last gene in this pathway. Importantly, what we observe is an accumulation of methionine precursors, homocysteine and homoserine. And the effect of homoserine alone can recapitulate the effect of trimethoprim. So when we treat cells now with homoserine alone in the absence of trimethoprim, we see a similar level of induction of this malleolactone gene cluster. Moreover, the metabolome that's elicited in response to trimethoprim very much mirrors that seen with homoserine. Furthermore, if we generate a MET-X knockout, which leads to accumulation of homoserine, we now see that in the absence of trimethoprim, this mutant has a malleolactone positive phenotype, and then with trimethoprim, the levels of malleolactone are further increased. And so the mechanism that's sort of emerging is one where inhibition or partial inhibition of DHFR leads to long-distance effects on this one-carbon metabolic pathway. Notably, the increase in homocysteine and homoserine induced then the silent malleolactone gene cluster. The mechanism by which this happens is something that's not known, and that is something that we are still working on and trying to figure out. Okay, so I'll quickly summarize. I showed you today a new method that we've developed to activate silent biosynthetic gene clusters, and the mechanism that is involved, at least in one case with Burkholderia and trimethoprim. That's an ongoing project. Ours hasn't been the only method. Other methods have been developed as well to activate these silent gene clusters. And I think together now, the stage is set to try to figure out what all these gene clusters are doing in these organisms, what their structures are, whether they'll be useful to us, and ultimately what their roles are for the microorganism. What we have found is that sub-inhibitory doses of antibiotics play a key role in this process, while high concentrations of the same ones kill. This is a property known as hormesis, and Julian Davies has pointed this out for bacteria. And again, this is with respect to secondary metabolism, a new subfield that we're currently investigating. And I think it'll be interesting to see what the sort of molecular details are between cell death and metabolism stimulation at high and low doses. And lastly, I hope what you take away from this talk is that there's still a lot of chemistry and biology that remains to be learned by studying cryptic biosynthetic pathways in bacteria. With that, I'll quickly summarize all the people that contributed to this work. I've had a fantastic group of former and current lab members. Danan, Yihan, and Bethany worked on the Heights approach in Borrelia. Chen, Fei, Behnam, and Kyuho worked on the Streptomyces heights and imaging mass spectrometry. And then Anran, Paul, and Danan worked on the mechanism of trimethoprim in Borrelia. I'm thankful for all of their efforts. Thankful to Dr. Kate Davis and Phil Jeffrey for the crystallography collaboration, these sources for funding, and again, Pete and Michael Gilmore for inviting me to speak today, and all of you for attending. I would be happy to answer any questions.
Wonderful. Thank you so much. Let me go ahead and there we go. All right.